So welcome to Chapter 12 of A&P 1. Today we're talking about nervous tissue. So the nervous system is one of the primary systems or functional systems in the body, and it's going to be one of the two main systems that helps to maintain and regulate homeostasis, and it's going to help to keep kind of control of conditions and maintain a healthy range for various parts of the body. The nervous system is divided into two systems. We have our central nervous system and the peripheral nervous system. The central nervous system is going to be composed of the brain and the spinal cord, and the peripheral nervous system is basically all of the other nervous tissue in the body. So anything that's not in the central nervous system. So all of the nerves except for the brain and the spinal cord. So we're going to start with the peripheral nervous system. The components of the peripheral nervous system are going to include cranial and spinal nerves as well as their sensory receptors. So a nerve is simply going to be a bundle of hundreds or thousands of axons, of nervous axons, plus it's any kind of associated connective tissue and blood vessels that are going to lie outside of the brain and the spinal cord. So like we mentioned, the nerves of the peripheral nervous system are divided into two groups. We have our cranial nerves. There are going to be 12 pairs of these nerves, and they're going to emerge from the brain. And then our spinal nerves are going to be the nerves that emerge from the spinal cord. And there are 31 pairs of these, and so there's a lot more. The sensory receptors are going to be structures of the nervous system that are there to monitor various changes in the external or internal environment. So these are going to be things like touch receptors in the skin, photoreceptors in the eye, the olfactory or smell receptors in the nose. All of these are going to be different types of sensory receptors. There are also other types of receptors too, things like baroreceptors to measure or monitor blood pressure, etc. So there are a whole lot of these sensory receptors that are really important for detecting these changes. So the peripheral nervous system is divided into two main groups. All right, we've got our ingoing group. This is the sensory division or the afferent division. This is going to convey input into the central nervous system from those sensory receptors in the body. All right. There are two different types or two different divisions of the sensory division. We have our somatic senses. These are things like tactile senses, thermal, pain, proprioception, all of the things that are sort of used by your body to determine or to keep an eye on your surroundings, your external environment. We also have special senses. This is part of the sensory or afferent division. These are going to be receptors that are going to convey input into the sensory nervous system, things like smell and taste, vision, hearing, and equilibrium or balance. The second division of the peripheral nervous system is the motor division or the efferent division. This is going to convey output from the central nervous system to effectors, whether it's muscles or glands. So our afferent system or division is going to convey input to the nervous system, and then our efferent division is going to convey output to the central nervous system. So our motor division, or our efferent division, is again divided up into two groups. We have our somatic nervous system. This is going to be what's responsible for conveying output from the central nervous system to the skeletal muscles. This is how you tell your body to move, to walk or to stand up straight. This is all done voluntarily. So it takes a certain level of conscious thought. The other division of the motor division of the peripheral nervous system is the autonomic nervous system. And this is going to convey output from the central nervous system to the various smooth muscle, cardiac muscle, and glands of the body. And this is typically involuntary. So this is going to be what's responsible for controlling things like heart rate, blood pressure, etc., various bodily secretions. So this is not something that we have to actually think about. This is an involuntary sort of event or an occurrence. So our autonomic nervous system then gets broken down even farther into three different individual systems. We have the sympathetic nervous system, the parasympathetic nervous system, and then the enteric nervous system. So the sympathetic nervous system is fight or flight. The parasympathetic nervous system is stuff like rest and digest, to like break down your lunch and stuff. And then our enteric nervous system is going to help to regulate the activity of the smooth muscles and glands that are found in the GI tract. So things that are going to actually involve digestion. So those are our three groups, then, our three systems in the autonomic nervous system, which are all part of the motor division of the peripheral nervous system. So here we have a nice little graphic here, looking at basically the breakdown of our nervous system here. So we have our central nervous system consisting of the brain and the spinal cord, like we said, and then conveying output is going to be our motor nervous system, and then anything that's going to convey input into that central nervous system is our sensory input, our sensory division. And these two divisions, the sensory division and the motor division, are going to make up our peripheral nervous system. Okay, so the sensory division, like we said, is responsible for detecting things like somatic senses and special senses. And that information or stimuli is going to be delivered to the central nervous system, right? Then our motor, or our output, our motor division, which is our output, then is going to receive information from the central nervous system, and it's going to stimulate reactions in one of these various options here. So our somatic nervous system is going to be responsible for any kind of voluntary movement. And then our autonomic nervous system, like we said, is broken down into three different divisions. We have our sympathetic nervous system and our parasympathetic nervous system. For our fight or flight and rest and digest, this is going to control the smooth muscles, cardiac muscles, and the various glands of the body. And then our enteric system is this third division of our autonomic nervous system. This is responsible for the smooth muscles and the glands of the GI tract specifically. So, looking again more specifically at the autonomic nervous system, the motor parts of the ANS are going to be divided into the sympathetic and parasympathetic nervous systems, like we mentioned. So effectors for this are usually going to receive innervation from both of these branches. So smooth muscles, cardiac muscles, glands are receiving innervations from both of these branches, both parasympathetic and sympathetic systems. And usually these two branches are going to have opposing actions. All right, so our rest and digest versus our fight or flight instincts are going to have sort of opposing actions, but are still going to be working on or innervating the same effectors. So those smooth muscles, those cardiac muscles, and the various glands. Our third branch of our autonomic nervous system is the enteric nervous system, or our ENS. This is going to consist of neurons in the enteric plexus that are going to extend the length of the GI tract. So a plexus is basically just this big kind of overlapping bundle of nerves that are responsible for innervating similar tissues or related tissues. So in this case, the enteric plexus is responsible for innervating the nerves of those enteric plexus is responsible for innervating various parts of the GI tract. So the enteric nervous system is going to help to regulate the activity of smooth muscles and the glands in the GI tract. So it's going to be responsible for kind of controlling peristalsis and the secretion of various digestive enzymes, etc. Also, the ENS is going to function independently, but it can also communicate with and can be regulated by the other two branches of the ANS, so our sympathetic and parasympathetic nervous systems. So while our enteric system does function independently, it's responsible for maintaining the various digestive functions of the GI tract. It can receive stimulation from those other two. So if for some reason there's a very high stress situation, the sympathetic nervous system is going to communicate with our ENS, and it's going to sort of slow down the actions of our enteric nervous system so that we don't need to be devoting as much energy, metabolically speaking, to digestion when we need to be potentially diverting that energy to preparing to run away. So they kind of overlap there. So the sensory neurons of that enteric nervous system are going to be responsible for monitoring chemical changes within the GI tract. And they're also going to monitor the stretching of those walls. However, our enteric motor neurons are going to govern the contractions of the various GI organs and are going to activate the various endocrine cells that are located in that GI tract. So, there are three main functions of the nervous system. All right, there's going to be a sensory function. This is where we're receiving sensory input information. Then there's going to be an integrative or processing function where your brain and the rest of your body decides what to do with that sensory input. And then there's our motor and our output function, so our response to that stimuli. So for our sensory or input function, this is going to be responsible for sensing changes in the external or the internal environment through our various sensory receptors. And it does this via sensory neurons, which we'll talk about later. Then once that sensory input has been detected and moved or brought up to the brain, essentially, the integrative or the processing function of the nervous system is is going to take over. This is where it's the body is going to analyze that incoming sensory information. It's going to store some aspects of that input, and then it's going to make a decision regarding an appropriate behavior. So this is done by the integration or inter or association neurons. So this is sort of where we're deciding what to do with our sensory input that we've just received from our sensory receptors. Once that that information, that stimuli, has been processed by our integrative functions, then we're going to have some sort of output. This is usually a motor output. So this is going to respond to stimuli by initiating some sort of action, by activating effectors through the cranial or the spinal nerves. And this is done via those motor neurons. Right. So let's look then at the histology of the various nervous tissues in the body. So, nervous tissue. Nervous tissue is going to be comprised of two main types of cells. We've got our neurons. These are going to be what's responsible for providing most of the unique functions of the nervous system. And then we have the neuroglia. These are sort of like the backup singers of the nervous tissue. These are going to provide support, nourishment, and they're also going to protect those neurons. So both of these are going to differ structurally depending on whether they're located in the central or the peripheral nervous system. So neurons are going to have a couple of different characteristics, and the first of which is that they're electrically excitable. And this is simply the ability to respond to stimuli and to convert it into an action potential. So a stimuli is going to be any change in the environment that's strong enough to initiate an action potential. And this is going to be detected usually by our sensory receptors. And then action potentials, sometimes called nervous impulses, are going to be electrical signals that are going to propagate along the surface of the membrane of a neuron. So these action potentials are going to begin and they're going to travel along our neuron due to the movement of ions between the interstitial fluid and the inside of that neuron through these very specific ion channels that are found in the plasma membrane of the neuron. So let's look then at the various parts of the neuron. There are three main parts to each neuron. First off, we have our cell body, which you can see here. This is kind of big blue area right here. This is going to contain the nucleus, the lysosomes, the mitochondria, the Golgi complex, all of the various cellular parts of the neuron, as well as cytoplasmic inclusions such as lipofuscin, chromatophilic substances, and neurofibrils. And we'll talk a little bit more about what those things are in just a minute. The second part of the neuron are going to be the dendrites, and you can see these kind of spiky projections coming off of the cell body. Those are our dendrites. These are going to be the receiving or input portion of the neuron, and there can be a lot of these. So these are responsible for detecting a lot of the stimuli. And then last but not least, we have our axon, which you can see down here coming off of our cell body. This is going to conduct nerve impulses from the neuron to the dendrites or to the cell body of another neuron, or to an effector of an organ of the various or to an effector organ that's somewhere else in the body. And there's usually only one axon per neuron. So, like we said, things are named differently or are going to look different depending on whether they're in the central nervous system or in the peripheral nervous system. So a ganglion is going to be a collection of neuron cell bodies that's going to be outside the central nervous system. So ganglion is a collection of neuron cell bodies in the peripheral nervous system. Okay. So let's then look at the cytoplasmic inclusions of the neuron that we mentioned previously here. These are all found in the cell bodies of the neuron. So our first one is going to be lipofuscin. And this is a pigment that's found in aging cells that's going to occur in kind of clumps of yellowish brown granules in the cytoplasm. This is going to be a product of neuronal lysosomes and is going to accumulate as the neuron ages. So there doesn't really seem to be any kind of harm that's caused by this lipofuscin. It's just something that kind of occurs normally as the neuron ages. Next, we have our nycil bodies, also known as the chromatophilic substances. These are going to be orderly arrangements of the rough endoplasmic reticulum, and you can see them in this little image right here. These are our nycil bodies. So these are going to be newly synthesized proteins that are produced by the nycil bodies, and these are, So the proteins that are newly synthesized by these nycil bodies are going to be used to replace any kind of cellular component that's worn out or that is needed in the neuron, as well as materials for growth of the neuron, as well as regeneration to damaged axons in the peripheral nervous system. Our third type of cytoplasmic inclusion that's found in a neuron is called a neurofibril, and this is going to be found in the cytoskeleton of the neuron, and is composed of bundles of intermediate filaments that are going to be there to provide the cell shape and provide support for that neuron. So let's look then at the dendrites of a neuron. So like we mentioned, these are going to be the receiving or input portion of a neuron, and the plasma membrane of these dendrites, as well as the cell bodies, is going to contain numerous receptor sites for the binding of chemical messengers from other cells. A lot of the times these are neurotransmitters. So dendrites are usually going to be these kind of short and tapering and highly branched projections off of the cell body here. You can see that in this little picture here. And many neurons, the dendrites are going to form this kind of tree-shaped arrangement of processes that are going to extend from the cell body. So the cytoplasm of these dendrites are going to contain nycil bodies, as well as mitochondria and other organelles that are needed to maintain the functionality of the dendrite, or that part of the neuron. Okay, so let's then look at the various components of the axon. So an axon is a long cylindrical projection that is often going to join to the cell body in a kind of cone-shaped elevation. It's called the axon hillock, which you can kind of see here. So the part of the axon that's closest to the axon hillock is going to be the initial segment. And in most neurons, the nerve impulse is going to arise at the junction between the axon hillock and the initial segment. This is going to be an area that's called the trigger zone, and from which these action potentials are going to travel along the axon to their destination. So an axon is going to contain mitochondria, microtubules, neurofibrils, but it is not going to contain any endoplasmic or rough endoplasmic reticulum. So because this rough endoplasmic reticulum is absent, no protein synthesis is going to be able to occur in the axon. The cytoplasm of an axon is called the axoplasm, and it is going to be surrounded by a plasma membrane that's known as the axolemma. It's the same cytoplasm and plasma membrane as you'd see in any other cell, but it is specific. These names are specific to the axon, because everybody likes to be special and have their own names for things. So along the length of the axon, there are these little side branches, or can be these little side branches called axons collaterals. and these are going to branch off of the main axon. And these are going to typically branch off at a right angle to the axon. So this axon and its little axon collaterals are going to end by dividing into many of these little fine processes called axon terminals, which you can see down here, are axon terminals. All right. So let's talk then about slow versus fast axonal transport. So things being transported up and down that axon. So because some substances are synthesized or recycled in the neuron cell body, that are going to be needed in the axon or at the axon terminals, there are two main types of transport systems that are used to carry this material from the cell body to the axon terminal and back. We have slow and fast axonal transport. All right. So our slower system, as you would have guessed, is called slow axonal transport, and it's going to move materials at about a rate of 1 to 5 millimeters per day. So it's slow going. It's going to convey axoplasm in only one direction, and this is from the cell body towards those axon terminals. All right. This slow axon transport is going to be responsible for supplying the axoplasm to developing and regenerating axons, and it's also going to replenish the axoplasm in growing or mature axons. Our other type of axonal transport is fast axonal transport. This is capable of moving materials at a distance of 200 to 400 millimeters per day, and it uses proteins that function as these little motors to help move that material along the surface of the microtubules that are found in the neuron's cytoskeleton. So fast axonal transport is going to be able to move materials in both directions, both away from and towards the cell body. All right, this is different than what we saw in our slow axonal transport, which only moves in one direction. All right, so fast axonal transport that occurs in an anterograde direction or a forward direction is going to move organelles and synaptic vesicles from the cell body to the axon terminals. So this anterograde transport or direction is the same as what we would see in our slow axonal transport. All right, it's also present in fast axonal transport. Retrograde transport is only going to be found in our fast axonal transport, and this is moving in a backwards direction. So it's moving membrane vesicles and other cellular material from the axon terminal to the cell body that's going to be then degraded or recycled to be used elsewhere. So substances that enter the neuron at the axon terminal are also going to be moved to the cell body via fast retrograde transport. All right, so in fast axonal transport, materials can go in both directions, forward and backward. All right, in an anterograde or retrograde manner. All right, and in slow axonal transport, it's only moving in an anterograde direction. So it's only moving forward. All right, it's a big difference there. So let's look at the structural classifications of neurons, because like everything, science likes to classify and group various types of structures. So neurons can be classified based on the number of processes that extend from the cell body. All right. So a neuron can be a multipolar neuron, and it's going to have several dendrites and one axon. All right. So here we've got this. We've got our lots and lots of dendrites and one axon. This is a multipolar dendrite. All right. So this is going to be what most of the neurons in the brain and the spinal cord, as well as all of the motor neurons, are going to look like. They're going to be multipolar. Our next type of structural classification of neurons is the bipolar neuron. And this is going to have one dendrite and one axon. All right. So this is going to be found in the retina of the eye, as well as in the inner ear, as well as the olfactory area of the brain. All right. So we've got our cell body here, and one axon with multiple dendrites coming off of it. All right. So here we've got our multiple dendrites. And then our third type of neuron, structural classification of neuron, is the unipolar neuron. All right, and this is going to have dendrites and one axon that are going to be fused together to form a continuous process that's going to emerge from the cell body. All right, so here we've got our cell body and our axons and dendrites that are fused together. Okay, these dendrites are going to be are going to make up most of the sensory receptors in the body. So most of our sensory receptors are unipolar neurons. All right. So just some examples of dendritic branching. So some neurons are going to be named after the pathologist that discovered them, or based on certain aspects of their appearance, because they don't really fit the other kind of classifications necessarily of our other structural classifications there. So things like a Purkinje cell, which we see here. These are going to be the sole output neurons for the cerebellar cortex and are going to play an important role in things like coordination, control of the body, as well as learning new movements. Okay, and then our pyramidal cell is named after the shape of the cell. These pyramidal cells are found in the cerebral cortex as well as in the hippocampus and in the amygdala. And they're going to serve as the primary excitation units for the mammalian prefrontal cortex. So a lot of decision making, higher functioning kind of decision making is going to be carried out by these pyramidal cells. All right. So again, we've classified our neurons via structure, so we can also classify them functionally. Neurons can be classified based on the direction of the nerve impulses the direction of which the nerve impulse propagates with respect to the central nervous system. All right. So we've kind of mentioned this before, but it bears mentioning again. For our functional classifications here, we have our sensory or afferent neurons. These are moving in a forward direction towards the central nervous system. All right. So here we have our sensory neurons, or afferent neurons. These are going to contain sensory receptors that you see down here. At their distal end, this is usually going to be dendrites, or they're going to be located just after the sensory receptors that are a separate cell. The action potential is going to be conveyed into the central nervous system for these sensory afferent neurons. All right. Next, we then have our motor or efferent neurons. These are going to be conveying action potentials away from our central nervous system. They're going to be transmitting those action potentials to the various effectors cells, things like muscles or glands, that are going to be in the peripheral nervous system through the cranial or spinal nerves. All right, so these are moving information away from the central nervous system. All right. And then kind of in the middle here, we have our inter or association neurons. All right, these are mainly going to be located within the central nervous system itself. And these are going to be kind of the link between our sensory and our motor neurons. So this is where the processing is going to take place for our sensory, or for the processing of the various action potentials is going to take place. These inter or associated neurons are responsible for processing incoming sensory information from the sensory neurons, and then are going to elicit some sort of motor response. All right, so it kind of works in a cycle. Some sort of stimuli is detected by our sensory neurons. It's brought into the sensory central nervous system, where it undergoes some sort of processing by our association neurons, and then that association neuron is going to sort of decide what kind of response needs to be elicited by our motor neurons, which are transporting information away from the central nervous system. All right, so sort of a U-shaped cycle here. So we've talked then about neurons. The other type of cell of nervous tissue cell that's found in nervous tissue is called neuroglia. These are specialized tissue cells that are going to do a variety of things, including supporting the various neurons. They're also going to be responsible for attaching the neurons to blood vessels so that they can receive appropriate nourishment and waste management. These neuroglia are going to produce the myelin sheaths that's found around axons, which we'll talk about here in just a minute. Neuroglia are also going to carry out phagocytosis to sort of keep everything clean and tidy. These neuroglia are not electrically excitable cells, so that is a big difference between neurons and neuroglia. They are not electrically excitable. And neuroglia are going to make up about half of the volume of our nervous tissue. So there's a lot of them, and these neuroglia are capable of multiplying and dividing. There are 6 kinds of neuroglia all total. 4 are found in the central nervous system, and then 2 are found in the peripheral nervous system. So let's look then at the neuroglia that are found in the central nervous system. All right, our first ones are astrocytes, and they're named this way because you can kind of see they look kind of like stars. I think these are really pretty to look at, especially when they're stained like this. Anyhow, these astrocytes are going to contain microfilaments that are going to give them a great deal of strength. This is going to allow them to be really useful in supporting the neurons. So the processes of astrocytes are going to wrap around blood capillaries that are responsible for isolating neurons in the central nervous system, and it helps to protect them from potentially harmless substances. And it does this by secreting chemicals that help to maintain this kind of uniquely selective permeability that's characteristic of the blood-brain barrier. So these astroglia are what's really responsible for the forming of the blood-brain barrier. So in the embryo, astrocytes are going to secrete chemicals that appear to help to regulate the growth, migration, and the interconnection among the neurons in the brain. So these are really important when you're developing. And then astrocytes are also going to help to maintain an appropriate chemical environment for the generation of nerve impulses. These astrocytes may also play an important role in learning and in memory by influencing the formation of neural synapses. So when you're a baby, there are a lot of neural synapses that are being formed by these astrocytes. And as you grow in age, you undergo something called neural pruning that kind of clears up and tidies up those neural synapses so everything's a little more streamlined. The next type of neuroglia that we have in the central nervous system are the oligodendrocytes. These are going to kind of look like astrocytes, but they're smaller and they have fewer processes coming off of them. So their processes are responsible for forming and maintaining the myelin sheath that's going to be around multiple central nervous axons, central nervous system axons. All right. So a myelin sheath is going to be a multi-layered lipid and protein covering that's going to be found around some axons, and it's there to kind of help insulate them. And it increases the speed of the nervous impulses that are able to be conducted there. So myelin sheaths are kind of like electrical insulation. It helps those. It helps that nervous impulse conduct down that axon more quickly. All right, and these are oligodendrocytes that are found in the central nervous system. Our next 2 types of neuroglia in the central nervous system are the microglia and the ependymal cells. So microglia are going to function as phagocytic cells. They are there to remove any kind of cellular debris that's going to form during any sort of normal neural development, as well as to phagocytize any sort of microbes or damaged nervous tissue. So they're kind of there to keep everything clean and tidy. And then our ependymal cells are going to be sort of a cuboidal or columnar shaped cell that are arranged in a single layer that are going to possess microvilli or cilia. These are going to line the ventricles of the brain and the central canal of the spinal cord, and they're going to monitor and assist the circulation of that cerebrospinal fluid. So these ependymal cells with their little cilia and microvilli are going to help to sort of circulate that spinal fluid or cerebrospinal fluid, just help everything flow properly. So we then also have the neuroglia that are found in the peripheral nervous system. We had the Schwann cells, which I think is kind of a funny name. But anyhow, these Schwann cells are going to encircle the peripheral nervous system axons and are going to be responsible for forming the myelin sheath around those axons. So each Schwann cell is going to myelinate only a single axon. And one Schwann cell can also enclose as many as 20 or more unmyelinated axons, okay? So here we see our myelin sheath that's being formed by those Schwann cells. And then we also can see how one Schwann cell can potentially enclose many or multiple of the unmyelinated axons, right? So our last type of neuroglia that's found in the peripheral nervous system are called satellite cells. These are our flat cells that surround the cell bodies of the neurons in the peripheral nervous system ganglia, and they provide structural support as well as regulate the exchange of materials between the neuronal cell bodies and that interstitial tissue, and that's really important, and we'll see why in just a little bit. So we've mentioned myelination a couple of times, so let's really look at what that actually is. So like we said, a myelin sheath is just a multi-layered sort of lipid and protein covering that's produced by either the Schwann cells in the peripheral nervous system or by the oligodendrocytes in the central nervous system. And this myelin sheath is going to surround the axons of a lot of different neurons. So this myelin sheath is going to help to electrically insulate the axons, and it's going to allow for an increased speed of nerve impulse conduction. So axons that do not have this myelin sheath are called unmyelinated. Right? So because myelin has a very high lipid content, it's going to end up looking, when we look at the various nerves or parts of the brain, it's going to end up looking white. So when we talk about the white matter in the brain, this is because the axons in that area of the brain are myelinated, and then the gray matter then is unmyelinated axons. All right, so that's just kind of an important little tidbit there. So let's look at the process by which Schwann cells form this myelin sheath. All right, Schwann cells are going to begin to form that myelin sheath around the various axons during fetal development. So each Schwann cell is going to wrap around about 1 millimeter of a single axon length, and it's going to do this by spiraling around that axon several times. Eventually, after multiple layers of this glial plasma membrane surround that axon, the Schwann cell's cytoplasm and the nucleus are then going to be found on the outermost layer. So here we can see our Schwann cell that's wrapped multiple times around our axon, and our little our nucleus and cytoplasm are going to form that outermost layer. The inner portion is going to consist of up to 100 layers of that Schwann cell's membrane, and this is going to be what forms the actual myelin sheath. So you can see that here. We've got our myelin sheath, and then our cytoplasm and the nucleus of our Schwann cell. The outer nucleated cytoplasmic layer of the Schwann cell is going to be what's responsible for enclosing or surrounding that myelin sheath, and this is called the neurolemma, which is going to be found only in axons in the peripheral nervous system. And this neurolemma is going to be really important for nerve regeneration, which we'll talk about in just a minute. So this is only going to occur in the peripheral nervous system. So when an axon is injured, the neurolemma is going to aid in the regeneration of that neuron by forming a regeneration tube that's going to be responsible for guiding and stimulating the regrowth of that axon. Gaps in the myelin sheath are called nodes of Ranvier, which you can see right here. And these are going to appear at intervals along that axon. So each Schwann cell is going to wrap around one axon segment between these little nodes of Ranvier here, or Ranvier. So here you can see. So we've got our Schwann cells that are wrapped around lots and lots of times to form our myelin sheath. And then in between these little Schwann cells, we've got our nodes of Ranvier. Right? So myelination in the central nervous system is achieved or performed by oligodendrocytes. These are the cells that are responsible for myelinating the parts of several different axons. So in the peripheral nervous system, each Schwann cell is going to wrap around and myelinate one small part of a single axon. In the central nervous system, with our oligodendrocytes, these are going to wrap around and myelinate multiple different axons. All right. So each oligodendrocyte is going to put forth about 15 or so of these broad, flat processes that are going to spiral around and wrap around our central nervous system axon, and these are going to form our myelin sheaths. So, like we mentioned, it's important. Big difference here is one oligodendrocyte can myelinate multiple CNS axons, in comparison to a Schwann cell, which can myelinate only one part of one single axon. So, in oligodendrocytes, a neurolemma is not going to be present because the oligodendrocyte cell body and the nucleus are not going to envelop that axon. So they're kind of out, sitting out here doing their own thing, with these projections coming off that are going to myelinate those axons, but that cell body itself of that oligodendrocyte is not actually on that axon. So nodes of Ranvier are going to be present with these oligodendrocytes, but there are going to be fewer of them. And axons that are found in the central nervous system are going to display to have little ability for regrowth after injury. And this is thought to be due in part by the presence of the neurolemma that's absent in these myelinations for the oligodendrocytes. This is also thought to be due, possibly this inability to regrow, is also thought to be due to the inhibitory influence that's exerted by oligodendrocytes on axonal growth in the central nervous system. So there's kind of a multiple layer there about why there's little to no regrowth after injury in the central nervous system. So just real quick, a couple of terms that just bear mentioning here. Like we mentioned, with the differences between the central and the peripheral nervous system, we call things have different names. So a collection of nervous tissue or a cluster of neuronal cell bodies in the peripheral nervous system is called a ganglion, right? Our cluster of neuronal cell bodies. But then in the central nervous system, our cluster of neuronal cell bodies is called a nucleus, all right? And then with regards to these bundles of axons, in the peripheral nervous system, this bundle of axons is going to be called a nerve, whereas in the central nervous system, a bundle of axons that's located in the CNS is called a tract. All right, so just some important terminology differences there between those two. So we've mentioned this white matter versus gray matter thing, so let's look at that a little more closely. So white matter is going to be composed of aggregates of myelinated processes. So it's this high lipid count that we find in myelin sheaths that's making it look sort of more white. And then in gray matter, this is going to contain nerve cell bodies, dendrites, as well as axon terminals or bundles of unmyelinated axons and neuroglia. So our gray matter is unmyelinated, white matter is myelinated. All right, important difference there. And you can see the difference in how this looks in actual human tissue. You can see the difference here in this white versus this gray matter here. So in the spinal cord, the gray matter is going to form a kind of H shape in this inner core, which you can see here. Looks like sort of a weird butterfly maybe. And it's going to be surrounded by white matter on the outside, and that's in the spinal cord. In the brain, it's going to be the other way around. We're going to have a thin layer, a thin outer layer of gray matter that's going to cover the cerebral hemispheres, and then on that inner layer is going to be our white matter. All right? So that is the end of part 1 for the nervous system lecture.